90,000 people are on a waiting list to get a kidney transplant, and 11 of them die every single day while they wait.
The solution some may find unusual: pigs.
Porcine kidney xenotransplantation represents a turning point by eliminating the organ shortage and offers a solution that is far better than dialysis. CRISPR is a method of gene editing that can be used to change the genetic material of pig kidneys so that when they are transplanted into humans, there is no immune rejection. This system is adapted from the CRISPR system used by bacteria to acquire viral immunity. So when a virus with an unknown genome attacks a cell, the Cas9 enzyme takes a portion of that viral DNA, breaks it down, and places it into the CRISPR locus of the bacterial genome. This makes the bacteria now immune to the virus. So when the virus attacks again, the CRISPR RNA, which is created from the CRISPR locus, guides the Cas9 enzyme to the target sequence in the viral genome. From this, the Cas9 enzyme can break down the viral genome and deactivate it so that the bacteria is not infected with the virus. Scientists can use this system to edit DNA. So scientists will create a guide RNA and insert that into the cell's genome along with the Cas9 enzyme. The guide RNA will attach the target sequence that scientists want to edit, and then the Cas9 enzyme will cut that target sequence off. Then scientists can use the cell's own machinery to edit or change the cell's genome.
So what are we actually doing with the CRISPR? Well, we're going to be trying to remove pig genes that produce specific sugars called xenoantigens. We're first going to look at alpha-gal. Now, pigs have an enzyme called GGTA1 that we do not. That's what produces the alpha-gal. Since pigs have the enzyme and we do not, we need to find a way to make that uniform. So what we're going to do is we're going to inactivate the GGTA1 enzyme. And we're going to do that by knocking it out. And that's what CRISPR does. It's a very similar story for our next gene, NEU5AC. That's the gene that we have. But pigs have a very similar gene, NEU5GC. Similar, but not the same. So when we see it, we're not going to be able to accept the kidney. What transforms our gene into their gene is an enzyme called CMAH. And using CRISPR, we can inactivate that enzyme and make it so that the kidney can be accepted.
Another way that scientists mitigate cross-species viral transmission in xenotransplantation is by inactivating the PER sequences found in the pig's genome. So firstly, scientists will locate the PER sequence inside the nucleus of pig cells. Then, using the CRISPR system mentioned previously, they will inactivate that sequence in the nucleus. Then they will transfer that nucleus into the nucleus of an egg cell, which is known as somatic cell nuclear transfer. They will then place that egg inside the surrogate mother, who then births newborn pigs that have inactivated PERVs but have normal physiology. The most risky form of PERV that can be transmitted to humans is PERV-AC, which is a result of recombination between the PERV-A virus and the PERV-C virus. It is twice as likely to infect humans as the PERV-A virus on its own.
Another way that scientists use CRISPR to edit pig genes are through adding human genes that make pig tissues less foreign. The main problem is our immune system. See, it sees pig tissue as foreign and it attacks it. Scientists are tackling this by adding select human genes to pigs. And in doing so, that helps their organs blend in with our biology. One of these genes is called CD59. It acts as a shield and it blocks the membrane attack complex. That's a part of our immune system that can destroy cells. With CD59, pig cells become much harder for human immune cells to kill. Another key change is that we have to add human thrombomodulin, which helps regulate blood clotting and inflammation. Pig thrombomodulin doesn't work well with human blood, but the human version actually restores the balance and reduces the rejection. So together, with the thrombomodulin and the CD59, these genetic upgrades move us closer to a future where life-saving organs can be grown within the animals and bridging that gap between scarcity and survival.